Hi, my name is Michelle Whirl-Carrillo. I am the PharmGKB director. I also am very involved in all the projects you see on your screen right now. So in PharmCAT, Cpic, and also to some degree in PharmVar. So I'm here today to talk to you a little bit about all four of these very important pharmacogenomic resources to give you an overview of what each one is and what makes them distinct, but also how they work together. It can be a little bit confusing. 3 out of the 4 of these resources have Pharm in the title, and so PharmGKB, PharmVar, PharmCAT can get confusing. People also are not sure what's the difference between PharmGKB and Cpic. So I'm hoping to address those issues today in my talk with you. So I'm going to just give you a brief overview and then go into each one of these resources. So PharmVar is really a nomenclature resource. The primary goal of PharmVar is to catalog all the different pharmacogenomic alleles and pharmacogenes. They do a lot of other important stuff too, but that's the major kind of goal of PharmVar. PharmGKB is a knowledge base whose goal it is to accumulate all the PGX that can be found from the peer-reviewed literature, from drug labels. PharmGKB looks at pathways. They create information from the stuff that they curate as well. And it's kind of like a centralized hub for pharmacogenomics. Cpic focuses primarily on writing genotype-based drug dosing guidelines. These are guidelines that clinicians can use actively in their clinic for a way to implement pharmacogenomics in their medicine practice. PharmCAT is a tool to help implement all of the information that Cpic is putting out. So it is able to take a patient's VCF file, so sequencing or genotype data, and then generate a report in the end with Cpic guidelines that are pertinent for that particular person. So that's just kind of an overview of the 4 different resources, and now I'm going to go into each one step by step. So I'm going to start with PharmVar. Again, this resource is very important for understanding the star allele nomenclature for pharmacogenes. That nomenclature is a little bit confusing to people that aren't really familiar with pharmacogenomics and the cytochrome P450s. The star allele nomenclature has existed for a long time. I'm just going to step back from 1 minute from PharmVar. It's been around since the 90s. It's pervasive in the literature. This is how pharmacogenomic studies are done. When they're genotyping patients, they refer to a star allele. Somebody is a CYP 2D6 star 1 / star 4. It was initially started around, well, it was started in the 90s, but it became formalized around 2000, 2002. The cytochrome P450 allele nomenclature database was started. It was really a website, flat files on a website. Other nomenclature efforts were started as well. And in 2017, PharmVar stepped up to replace the original cytochrome P450 allele nomenclature database. And they really took this job seriously and started developing a really strict set of rules for how the star alleles get assigned and so forth. One of the reasons why this is a screenshot of PharmVar. We're looking at an entry for CYP2C19. And so if you scroll down the page, you would see all kinds of star alleles. There's sub-alleles. It gets really confusing to understand what's going on. One of the reasons why PharmVar is so important is in pharmacogenomics and many of the pharmacogenes, including the cytochrome P450s, it's variation across the entire gene that's important. There can be multiple SNPs. These are screenshots. I'm not doing live demos today. I'm going to show you screenshots of these resources, but I do want you to get an idea of what it looks like when you're on the website. So imagine I'm scrolling down the page for CYP2C19. This is what it looks like below. Again, you have more star alleles. They go on and on. You can see that each star allele can have multiple variants in it. And the same variant could be in multiple star alleles, and one star allele can have multiple different, each star allele has a different combination of variants, but they're overlapping. And so that's why for cytochrome P450s and some other alleles like TPMT, some other genes like TPMT, we can't just refer to one variant because it's the combination across the gene that influences the function and the definition of that allele. So that's why the star allele names are so important. It's important to understand. They're used, like I said, all over the literature. They're also used in genetic reports, clinical reports for patients. That star allele nomenclature is used everywhere in commercial labs, clinical labs. And so it's critical to have a resource that can show you really succinctly and clearly the distinction between all the different star alleles for every gene. Now, I imagine I scrolled back up the page for CYP2C19, and above where all the allele definitions are, we have some important parts of PharmVar. PharmVar is great because it lets you switch your reference sequence that all these genes are mapped to. That was a huge undertaking. Before PharmVar started, everybody had to kind of do that mapping themselves. And some people might have referred to a variant in a star allele with based on one reference sequence, and somebody else might have been using the cDNA, and somebody else might have been using the, the location on the entire, on the chromosome. So PharmVar allows you to easily switch back and forth between the different reference sequences. And if you clicked on another reference sequence, it would automatically update the positions of all these variants. Very, very nice feature of PharmVar. Another cool feature of PharmVar is it lets you compare without having to scroll down to see what's the difference between all these alleles. So if I clicked on this compare view, and I could say, oh, I'm interested in all these different star alleles, and I press the compare haplotype button, it will give you a little table that shows you, okay, these are all the variants involved in all of these star alleles, and you can see like star 2 has multiple variants, star 3 has a couple, and then you can see this variant, oh my goodness, it's found in a bunch of star alleles versus this variant is only found in star 2. So so it's not found in any of the other alleles that I selected. They also give you some clues about these variants. This little symbol means that that variant is probably altering the function of that star allele. So you can see star 2 has a couple variants that are probably altering the function. So star 2 is not going to have the same function as the star 1. So there are some really cool tools in PharmVar, and I wanted to point out one other thing. We're going to circle back to Cpic later, but one of the ways that we're connected here, all these resources together, is that while PharmVar is the ultimate source for defining all the variants in each star allele, they also do display some function information that's generated by Cpic. Cpic depends on these star allele definitions, and PharmVar uses Cpic clinical functions to describe the variants. So PharmVar relies on every single gene has an expert panel. There's actually a lot of work behind the scenes that goes into trying to follow the rules to break out these star alleles, how you know when someone submits to your database, if it's a new star allele, if it's a suballele of an existing allele. So we have panels of people that work on every single gene. There are all kinds of designation criteria and README docs that they go through and they can break down for you what's a major allele, what's a subvariant, what are the rules for assigning these. So I'm not going to get into the nitty gritty there, but I want everyone to know that all that information is transparent. It's found on PharmVar. And if you have the patience to read through it, it will explain to you how the nomenclature is derived. Okay. So I'm not going to go into any more on PharmVar. And I'm going to switch over now to PharmGKB. So PharmGKB is the pharmacogenomics knowledge base. It was started in 2000 and is housed at Stanford University. And the goal, like I said, is to collect all the pharmacogenomics knowledge that they can find. And they curate this information both manually, and there's some automated processes as well. And then they disseminate it through the website. So PharmGKB thinks of their mission as this triangle. We start with primary information like peer-reviewed publications and drug labels, things that are just that exist out there. We take that information, those resources, extract information, the PGX information from them, aggregate it together, and our goal is to go towards clinical interpretation and clinical implementation. So I'm going to show you how we do that at PharmGKB. This is a screenshot of the homepage of the PharmGKB website. We have some quick entry points to some of our most commonly accessed types of information, but we also have a search box. So if you come to PharmGKB, you can also always put in any gene or drug of interest and find information about that particular gene, drug, or variant. And if you scrolled down the page, I don't have room to show everything on this screenshot, but down the page, we have a lot of intro materials, especially over here on this right-hand side. There's a whole section on what is PharmGKB, what are all the types of information that can be found on it. So if after this presentation, you want to know more, or you've forgotten what I've said, this is a great resource on the homepage. There's a button right below here that will review all different types of information I'm going to give you in this talk. So at the bottom layer of that pyramid, that triangle that I showed, we had peer-reviewed publications. So we take a publication like this one. This is just a paper that was published in New England Journal of Medicine. This is an older paper now, but on SLCO1B1 and myopathy in statins. So we read, the curators at PharmGKB read a paper like this, and they extract specific pieces of information and tools that are provided to them. They are able to build a sentence about what is going on between the variant in this gene in this paper with the phenotype and the drug. And so they build a sentence like this for every single paper that's read, every variant in the paper that's read. They're able to put a little bit of extra information about the paper in free text. This is all very well structured data, this sentence. We use standardized vocabularies. We have a standardized sentence structure so that these annotations from every paper can be aggregated. People can use computational methods to compare and contrast and aggregate these sentences. But we also have free text, like I said. We have what paper it comes from, and then we collect some other pieces of information, such as, the p-values that are reported for the association, other statistics on it, what type of study it was, and so forth. So this is what we call a variant annotation in PharmGKB. It's based on one paper, that paper that I showed you from New England Journal of Medicine. So this is just reporting what that paper says about that variant. But we have many papers sometimes about the same association, right? A lot of people might publish about that. So what we do is we annotate every single paper, but then we group them together and we come up with summaries. So we call these clinical annotations on PharmGKB. They are summaries of all the literature that we have curated about this particular variant with this drug and the phenotypic outcome. These are written as a like genotype-based statements. But if you scroll down underneath these statements about this variant with this drug, you would see all the papers. So this is the one I think I just showed you, but we have many, many, many others papers that were annotated about the same association in PharmGKB. So the point of this clinical annotation is to do an overview. Sometimes many studies are published and they all agree with one another. Those are beautiful times. So we have replication of the results, and then we have a lot of confidence in the fact that this association is a real association, been replicated in independent cohorts many times. Sometimes, though, you get conflicting information in the literature, right? So one paper might say that two things are associated, and another paper might contradict that. this happens as well. So when we write these summaries, we're trying to give you an overview of everything that we found about it. And so we give you a level of evidence to give you a visual cue right away if there's a lot of evidence all pointing in the same direction, or maybe the evidence is all over the place, or maybe there's only 1 or 2 papers out, and maybe it's only a case study. So, so in addition to being able to scroll down below and see all the supporting evidence for these statements, we put this level of evidence on here. And on our website, we also have a paper published about this. You can see our definitions for our levels of evidence. Basically, a level 1 is the highest level, and a level 4 means we don't have very much information about how sure we are about this association. And level 1A means that we know that a guideline has been published, or that FDA label has a recommendation for using this genotype. We know that that people are actually implementing on that. Okay, another aspect of PharmGKB is we also use drug labels, such as FDA drug labels, as input on PharmGKB. So this is a screenshot of an FDA resource that's out there, the Table of Pharmacogenomic Biomarkers in Drug Labeling. If you scrolled down this page, you would see a list of FDA drug labels where they say we have a gene stated on the label, which might be of importance for this drug. It doesn't mean a whole lot more than that, this particular table. It just means there is a biomarker on the label. So at PharmGKB, we take those labels and we read them, and we try to assess for people what's going on the label with respect to the biomarker. In some cases, the biomarker is necessary in order to prescribe a drug. You have to test for something. That is only true in a very handful of cases, and primarily cancer therapies, but they do exist. Some labels use a little bit more wishy-washy language, but they kind of recommend testing. And the bulk of the labels don't recommend testing either. They just say, this gene is associated with the pharmacokinetics or whatever of the drug. So PharmGKB came up with these labels. These were conceived of at PharmGKB and defined at PharmGKB. So I want to be clear, these aren't FDA kind of approved categories. So we have this information on our website if you want to read in more detail what the criteria is for each of these levels. But the point is, we assign each label that we read one of those levels. We also go into detail on the label and try to pull out, okay, does the label say I should actually change the dose based on a variant that a patient might have? Do they say a drug is not indicated for certain patients with certain genotypes? And then in some cases, they don't say change the drug or the level per se, or the dose per se, but they do say monitor these people more closely, or consider changing something. So we have a bunch of tags on PharmGKB that we defined, and that we add to these labels to try to give our users an idea of, okay, there's hundreds of labels on that biomarker list. What actually is going on with each label? So this is a screenshot of our landing page on PharmGKB for drug labels. We curate not only labels from FDA, but also other resources as well. EMA, we had a collaboration with Swissmedic, a group that looked at labels from Swissmedic. We also have Canadian labels and a collaboration for Japanese labels. Okay, but then for each drug along this side, we have, you can see in this column, if we have an FDA label curated about it, or a label from another agency. This tells you, okay, on this particular label, there is a variant that is required to be tested for before you can prescribe the drug. And then we have all these other tags that I showed you as well. So this is supposed to give somebody just an overview right away by looking generally, okay, what are maybe the drugs that are standing out on drug labels as have most important PGX information. But you can dive down into each one of these. You can click on one of these and get more information. So for example, if I clicked on the FDA label for phenytoin, I could see, I get a summary. I have a little excerpt about what we think at PharmGKB might be important for a clinician to know who's prescribing. So in this case, they say, you should consider avoiding this drug for a section of patients. They don't say anything about testing, so it's just actionable to us at PharmGKB, but we do point out to users that, you know, there is some guidance there on the label, even though there's no testing recommendation. So if you knew this, you could act on it. We also have, you can download from our website the actual label itself, where we've highlighted where we get our information from that we're displaying on the page. Okay, moving on from labels. We create pathways on PharmGKB based on the literature that we curate. So we make those variant annotations and clinical annotations I showed you, but we also try to build pharmacokinetic and pharmacodynamic pathways for drugs based on that information as well. So we make a pictorial representation of what's going on with the metabolism or mechanism of action for a particular drug. This is just a screenshot of a pathway for clopidogrel, a PK pathway. Oh, sorry, I'm trying to advance my slide. There we go. We also, if you scroll down, have a whole description. And actually, the description is most of the time a paper that's actually published, oftentimes in the Journal of Pharmacogenetics and Genomics. So most of our pathways are published as well. And you can have access to that paper from our website if you want to read more about a particular pathway that we have created. And then we have computational information available on the website too, that show exactly for every single piece of the pathway, what is the, you know, the literature reference that we're building this on. Every arrow has some evidence behind it, is what I'm trying to say. So we have those PMIDs are displayed, and these can be computationally operated on. Our GP, we have different files that that can be used in a computational manner, as well as just a nice display. So there's more information on PharmGKB than those types of information that I showed you, but you can read more about some of these if you go to the PharmGKB website. I just want to mention that we do write and publish summaries about pharmacogenes, also typically in the Journal of Pharmacogenetics and Genomics. And we also have prescribing information on PharmGKB that either come from published studies. Oops, I am sorry about that. Either published studies, so a single study, what they did in that perspective, a retrospective or prospective trial, and we also have genotype-based dosing guidelines from expert consortia that we curate and annotate on PharmGKB. Cpic, which I'm going to describe a little bit later, is one of them. But we also look at guidelines from other groups as well. There are other groups that exist that also write clinical guidelines about pharmacogenomics. I'm going to show you an example of this later on PharmGKB after I discuss Cpic. So just to give you an overview of PharmGKB, we have large amounts of information. We've been doing this a long time. So we have curated many drug labels, many peer-reviewed articles. We have thousands of clinical annotations. And so all that information is indexed and can be searched on PharmGKB using that search box that I showed you on the homepage. And then I just wanted to say one little thing about pediatrics in PharmGKB, because I know the Sanford group has a particular interest. It is true that there are not copious amounts of pediatric literature out there that we have found that focus on pharmacogenomics, but we do have you know, quite a few annotations that we have been able to create from literature about pediatric pharmacogenomics. They do exist on PharmGKB, and we are working right now on ways to highlight those on our website so that they're easier to access, and you can do a search on pediatrics. Right now, if you search pediatrics, you will not necessarily pull this literature back, but we're very close to being able to display that. So I'm hoping to highlight that on PharmGKB. We'll put it out on our blog when we have it ready. We're also looking towards highlighting pediatric information from those drug labels that we annotate. and about potentially creating drug-centered pathways where we take into account the expression at different age, at different time periods in fetal and child development, depending on when metabolizing enzymes are expressed. So that should be relevant to the pediatric community as well. Looking forward to that. Okay. I am going to switch over to Cpic now. So Cpic is the Clinical Pharmacogenetics Implementation Consortium. This project started as a collaboration between the PGRN and PharmGKB. And it started, I think, about 10 years ago now. So the guidelines that Cpic writes are designed to help clinicians understand how to use genetic tests. So we do not focus at Cpic on whether, giving advice on whether to test or who to test, but assuming that you have that information available to you about a particular patient, Cpic's goal is to explain to clinicians how they can use that information. In today's day and age of DTC genomics, some patients do have that information themselves and present it to their doctor. We collaborate with many academic hospital systems where they are starting to preemptively genotype patients as they come in. So in these cases, they can refer to Cpic guidelines for how to use the information that they already have on hand. Cpic is a global consortium. We have members from all around the world. A pretty large, it's way larger than 250 members now. That was a year or so ago that we hit that number and passed it. So Cpic tries to prioritize, well, they don't try, Cpic prioritizes which gene pairs get a guideline created by going through kind of this flow chart system. We look at if there have been other societies that have written some kind of guidelines, if there is external guidance from FDA or EMA or a Cpic member who has implemented a gene drug pair. We look at the PharmGKB clinical annotations, the levels of evidence that I mentioned. And we also take into consideration if other professional societies have written a guideline but don't recommend any action. So this information is taken into account, and then each gene drug pair is assigned a particular level as described here. And I will point out that an interaction between Cpic and PharmGKB is this information that Cpic uses to evaluate gene drug pairs gets pulled from the PharmGKB resource because we curate all this information at PharmGKB. So there's a dependency there. After a gene drug pair is decided to be very high level, it gets to level A or B, and we have a committee together that's committed to writing a guideline on it. We write and publish papers. Most of the Cpic guidelines, all to date, are in the journal Cpic. This is one example of a paper that came out recently about CYP2D6 and atomoxetine. So the front-facing view of Cpic's work are these manuscripts, but there's much work that goes on behind these manuscripts, behind the scenes. For every guideline, such as the guideline that I showed you, we start out by evaluating a ton of literature evidence. So this table scrolls down, down, down. These are all papers that have been looked at. We start with a huge literature review. We do a PubMed search, try to find everything we can, in this case, about the association between CYP2D6 and a drug like atomoxetine. Huge lit review is undertaken. We try to come up with major findings that these papers support, many times dozens or more major findings for any particular gene drug pair. Our author group reads those papers and ranks them and says for each major finding, yes, there's a lot of evidence. We use high, moderate, and weak. The SOPs used to evaluate the literature and create the guideline have all been published. You can go to the Cpic website to get those publications. And so we've published information about how we use this ranking system to rank the major findings. Then we use that ranking in order to come up with the actual recommendations for that gene drug pair. The first thing we have to do to get a recommendation is we need to link the genotype to a particular phenotype. So in this case, we've got all different kinds of CYP2D6 metabolizer status, so from ultra rapids to poor. With CYP2D6, it's a little bit of a special case. People use something called an activity score also in addition to the phenotype in order to rank and group together the diplotypes. So every Cpic guideline has a table like this where we have to link genotype to phenotype first. And then we have a second table where we connect the phenotype to the therapeutic recommendation. So Cpic guidelines are very specific. They give specific action to clinicians about what to do if their patient has a particular phenotype, which we trace back to genotype. And then I wanted to point out that each recommendation has its own strength, the recommendation strength. They're strong, moderate, and optional. So in order to actually be able to implement that in the clinic, we need to go back and Cpic and really give people the materials to be able to determine the genotype, to map to the phenotype, to do every step of that process along the way. So we create a bunch of supplementary files. This is one such supplementary file that Cpic and PharmGKB create together. And in this case, we're actually back to CYP2C19 and those star alleles I showed you from PharmVar. We show a slightly different representation, but this is based, these tables are based from the information that we pull from the PharmVar resource. We also align them to multiple reference sequences so that people can, no matter if they use cDNA or genomic DNA or whatever, they're able to, or RSIDs, they're able to identify each variant in every star allele. We also create tables that have every star allele and give a function for that star allele. So remember, so the star allele is a representation of all the changes along the entire gene, and those changes change the function of the gene in some cases. So typically star ones are normal functioning because they're your reference sequence essentially in most cases. But some star alleles are essentially dead, some have a decreased function, some actually have increased function compared to the star one allele. So this is actually putting together these tables and trying to define the function for each one of these alleles is a huge undertaking by Cpic. We spend a lot of time, again, reviewing the literature for evidence of how the variants in each allele affect the function of the gene. And we have, if you were to scroll, you would see copious notes about how we made the decision about how to assign the function for each allele. This is done again as the author group for each guideline. Not only do they review the literature about the association with the particular drug and that drug response, but overall literature about for each allele, what happens, what has been reported as the outcomes for these star alleles. Some of these are in vitro studies, some of these are in vivo. We have a whole SOP that's available on the Cpic website to show what all is taken into account, how we make the decision to assign the function, but a lot of work goes into that. And these functions are what it gets shown back on that PharmVar website that I showed you at the very beginning. We also come up with tables for allele frequencies. These, because they're combinations of variants across the entire gene, they're not as simple as like going into Nomad or 1,000 genomes and just picking out one variant and reporting the frequencies. So we are really dependent upon the literature where people have said, I assayed for this particular allele, and I found this frequency in this population. So again, a huge, if you go to the references tab on this big Excel spreadsheet that's available on the Cpic and PharmGKB websites, you can see all the different literature that we've evaluated to try to calculate allele frequency. We tried to split it up across different global populations because frequencies will vary. Of course, these are just averages. In some cases, there's really nothing known. If you have a blank here, it means that we really don't know what the frequency is. We couldn't find anything reported in that global population with that particular allele. So it just shows you also like where maybe we're lacking information about frequency. Okay, we provide files from Cpic to map from the diplotype to the phenotype. We have EHR priority result notation. We provide CDS, clinical decision support, example text, and example EHR workflows. All this information by Cpic is available on the Cpic website. So please visit it, and you can really dig down into this in more detail. And I just wanted to say, just mention one thing that, oh, I'm sorry. Yes, pediatrics and Cpic. That's true. Most guidelines have pediatric considerations. It depends drug by drug, but for most, while the evidence for actionability in pediatric populations are scarce, evidence is scarce. For the majority of cases, there's not really evidence that you would need to do something different either. So, but this is a focus of Cpic as well. And as I said, we try in every guideline to find some evidence of how to implement this in the pediatric population. And when possible, in some cases, we actually have a lot of information and we're able to get a whole separate table for the pediatric population. And yes, this is where I wanted to mention the Cpic guidelines that I just showed you from the Cpic group can be found back on the PharmGKB website as well. We have the Cpic guidelines as well as the guidelines, like I said, from the Dutch working group. There's a Canadian group, there's a French group that have a few. Yes, if you go to, on any drug, you go to the prescribing info section, you can find these guidelines. And on PharmGKB, what we have done is we have created a little tool with a drop-down box so that you could select the alleles. Like if you wanted to see what's the recommendation for a particular diplotype, we kind of try to do all those calculations for you on the back end. So all those tables I showed you about with the function and the mapping to the diplotype, that's on the back end there. And so if you pull something down, then you can see, oh, here are the implications. Here's all the information from table 2 in that guideline that I showed you from from Cpic. And then I just also wanted to mention that we do have some videos as well, where we try to summarize in just a couple minutes the crux of what is in the Cpic guideline. Of course, we recommend reading the guideline in full for all of the little details that matter. Okay, and the last resource that I'm going to talk about briefly is PharmCAT. Okay, so the motivation for creating this tool was we have all this information I've shown you, right? We know that there are allele definitions. You have to have certain variants in order to even assign someone a certain allele. There's function information to take into account. There's a mapping to the phenotype. There's the recommendations for Cpic, but how do you get the recommendations if your patient comes in with some genotypes? So we want to be able to provide that for people. So we know that there have been multiple studies. This is just one from Geisinger where they found, 96% of their subjects had one or more variants that was actionable in Cpic. So, this is not rare to have a patient that may have a variant that is actually actionable. We know that the Cpic guidelines exist. They give us copious amounts of resources to use to be able to, to be able to go from the genotypes all the way to the recommendation. they all exist, but somebody has to implement that. It should be straightforward because all those exist, right? So all those tables exist. It should not be that hard for someone to just go from point A to point B, you would think. However, the actual assignment of the haplotype and diplotype can be tricky because of those overlapping variants that I mentioned way back at the very beginning in PharmVar. The same variant can be in multiple star alleles, and then each star allele has its own combination. And so if you're only looking at certain variants, you might get confused as to what star allele you're dealing with. And if you don't get that haplotype and diplotype assignment done correctly, that affects the phenotype of your patient and ultimately the recommendations. It can affect that. So I'm going to give you a brief example. I'm trying to show you, these are DNA sequences. These are positions along the sequence, and these are different alleles. This combination of bases at these positions results in a CYP2C19 star 17 allele. This combination is a star 4, and this combination is a suballele of star 4. Okay, but there's these two positions that really matter. So you can see this one has a TA, this one has a CG, and this one has a T and a G. So it's a little bit like star 17 and a little bit like the regular star 4. So if you just have a star T, a T and an A in this particular combination of alleles, you are a star 17, which has an increased function. If, however, you have a G at this other position, you are a star 4, and that has no function. This is an upstream variant that turns up transcription. And so if you have this T, you're making a lot more. So that's why you have an increased function. However, this G is a killer. If you have this G, you have ruined your start codon for, for the gene. So no matter if you have a T upstream or not, you can't make any of that enzyme, right? Okay, so that's why this is a no function. So if you have these together, like I said, even though you have this upstream variant that turns on transcription, you're not making any protein. So this star 4 suballele is still no function. Okay, why does this matter? Okay, so if you have just the T variant, you would be assigned a star 1, star 17 diplotype, and people would say you're a rapid metabolizer phenotype. Okay, but if you have the G, then no matter how you look at it, you're not a rapid metabolizer. You're at best an intermediate metabolizer phenotype. You could have, there's a couple ways to view this. If you know that these are together on one chromosome, then you're a star 1, star 4 suballele. If you don't know whether or not these are on the same chromosome, you'd be star 17, star 4. That's still an intermediate phenotype. So the difference is, why does this matter? If you are a star 17, people are going to tell you to use less of a drug. The Cpic guidelines will tell you to do one thing for rapid metabolizer, but an intermediate metabolizer has a completely different therapeutic recommendation. So misassigning your patient in this case could change from, you might have misassigned an intermediate metabolizer to a rapid metabolizer status, and therefore your therapeutic actions are not appropriate for that patient. So I hope I didn't lose you too much in that kind of convoluted example, but what I want you to take away from this is that the assignment of the haplotype and diplotype can affect phenotype, can affect what you do for a patient. So the best that we can do is be really transparent then with what tests are given, how a patient, what variants are assayed for, and how a patient is given a particular haplotype status. That's very important. A lot of clinical and commercial labs may make that information available in a very small footnote, and a lot of people don't know to even look for it. But what we're saying with PharmCAT is if the genotype information is there, you should use it. No matter what positions were looked at, use all of that information. And if anything is missing, you have to do the best you can do. This is true. We use genotype chips all the time. You have to just use what you can. But if you're missing critical information, you should at least say so. So PharmCAT basically takes a VCF file in and issues a report out at the end with Cpic recommendations. You take the VCF file. This is an example of what a VCF file looks like. There's a lot of expectations for how this VCF file is formatted in PharmCAT. You can read all about it online. We take the definition files that I showed you from Cpic that were based on PharmVar. Those are used in PharmCAT, definition files like this. We use that to generate the diplotypes. We have a whole software package that does that. We have a separate way for calling CYP2D6 genotypes. That's a whole different can of worms. I'm not going to go into that here. We also take all the guideline recommendations from Cpic. We right now use the PharmGKB database and interface in order to get those annotations. We will soon be using the Cpic database itself. We have a whole bunch of extra information that's generated in PharmCAT by our scientists that are part of this package. We put it all together to generate a report. And the report at the very end of PharmCAT is a report for one patient that says, based on what was provided in their VCF file, what their diplotype may be, what the functionality of the alleles, their phenotype would be. We also want to tell you upfront if we were missing information, and then we have the drug report, like what are the Cpic recommendations, like don't use this drug, etc. This is a quick overview. If you went down the report, you'd see information in more detail. We give you drug by drug what the Cpic recommendations are for that particular patient. All the information about what variant positions were available to us when we made that call, if we were missing information. In this case, this patient was called a star 1 because it was really exomes. We didn't have access to that star 17 SNP that I said was upstream that caused an increased function. If you're doing exomes, you'll miss that star 17 in CYP2C19. So we give a warning, hey, star 17 wasn't there. Therefore, we're saying that your patient is a star 1 normal metabolizer. However, we couldn't tell you they might be a rapid metabolizer, but we couldn't determine that because that variant was missing from your data set. This is just an overview of the schema of PharmCAT. We have papers that are out that you can read that go through that whole thing. Okay. So there was a ton of information, I know, and I just want to spend a couple minutes just kind of reviewing what these different resources are and how they interact with each other. So remember, PharmVar is our nomenclature resource. We invest a lot of time at PharmVar trying to make sense of all the different information that gets submitted. PharmVar actually has information submitted to them from research, clinical, and commercial labs. People submit the sequences that they found, and then we also get information from the literature at PharmVar and use that to follow our rules about how the star alleles are created to assign the nomenclature for those alleles. PharmGKB takes a lot of PGX publications as well as expert guidelines and drug labels. Those are the main inputs to PharmGKB. For content, Cpic is based almost entirely on the peer-reviewed literature. Those extensive literature reviews we do at each step are based on the peer-reviewed literature. And remember, PharmCAT is a tool that basically takes information from these different resources and puts them together and allows you to take one person's or a group of people, but one person you care about at a time, information, and come out with what you could do based on these resources for that particular patient. So there's a lot of interaction. PharmVar's alleles are highly used. PharmGKB and Cpic are basically dependent on PharmVar's definition of those star alleles because the literature refers to them and the drug labels refer to the star alleles copiously, like I said, over and over again. And so PharmVar is our definitive source for what is the definition for each one of those alleles that people are talking about. Cpic also supplies PharmVar with that function information per allele. PharmGKB and Cpic are very much interdependent. As I said, Cpic started as a partnership with PharmGKB and the PGRN. Before there was even a Cpic website, we displayed Cpic information on PharmGKB. Cpic is its own entity. PharmGKB, as I said, strives to be a centralized resource of everything pharmacogenomic that we can find. We evaluate many more drug gene and variant drug pairs and associations than Cpic does, but on maybe a more basic level. So PharmGKB can inform you extensively on what information, what has been published about the interactions between genes and drugs, but we don't give you guidance on what to do. We tell you what could be done. Like, for example, patients with this genotype might experience, toxicity or lowered efficacy or whatever with this drug, and we can show you all the evidence we have behind that statement. But Cpic is the group, that's the expert group that gets together and writes actual guidance for clinicians about if you have a patient with this genotype, we recommend you do this. But as I said, they go back and forth. PharmGKB uses Cpic material. PharmGKB shows Cpic guidelines. Cpic uses PharmGKB criteria to help assign the gene drug levels that they do, help select which gene drug pairs they're going to write guidelines about. And then PharmCAT is this tool at the end that is in fact dependent on all of these resources. It's dependent on PharmVar, but via Cpic and PharmGKB. And so again, this is a tool, not a resource per se, like these other other groups. And just note, PharmCAT output could potentially be used in labs. So kind of full circle from research and clinical commercial labs submitting to PharmVar. In the end, they could use the output from PharmCAT theoretically. And one more thing I wanted to point out is that both PharmGKB and Cpic submit information to both the ClinVar resource, and now we have a partnership with ClinGen as well. And ClinGen ultimately will be displaying links back for PGX to PharmGKB and Cpic. So that's yet another kind of interaction between these big resources that I described to you today. And I'd just like to finish up by thanking everybody involved in all of these projects. There's many people involved in all of these projects to make them work. And there is a lot of overlap too, if you read the names. Terry Klein is co-PI of PharmGKB, Cpic, and PharmCAT. And works very closely with the PharmVar group as well, and as we all do at PharmGKB and Cpic, we all kind of work together. Does anybody have any questions?
Yeah, this is Stephen Mine from Wisconsin. Very nice summary for somebody who's not that much of an expert on pharmacogenomics. I did have one question. You made a very good point about the problem of distinguishing phase in diploid humans. And I'm just wondering, given that most people are doing short read sequencing for their patient analyses, how do people in pharmacogenomics get around that and establish phase? Because it seems from a clinical recommendation standpoint, you would really want to know phase for these haplotypes.
Yes. Ideally, you would. However, many times you don't. As you mentioned, you have short reads in some cases. Some people just use SNP chips as well. And of course, there's no phasing there. What is typically done is there are a couple things. One, people limit the variants that they look for. So for example, that star CYP2C19 star 4 variant in the start codon that I mentioned is actually not typically assayed for. So most of the time when people see the upstream variant, they're just going to call the person a star 17. It's not ideal, but the reason why this works most of the time is allele frequencies. So to be honest, that upstream variant is very common. And the change in the start codon, that star 4 allele, is rare. And so most of the time, you're fine if you don't look for that star 4. And so that's why things like the frequency tables come into play. That's why people look for those, because it can give you some reassurance that, okay, if I don't need to look for, 5,000 variants, I can just limit it to a doable handful. But at the same time, I have to put out the warning that when we don't assay over and over again, we don't really know what the frequency is. So like those blank spaces on the frequency table are there because sometimes we just don't look for certain variants. We don't look for certain alleles. And so we don't have good frequency information about them. So people don't face is the bottom line. A lot of times they don't. They just rely on the fact that frequency-wise, they're probably going to be right if they make an educated guess.
Hi, Michelle. There are a few questions in the chat. If you could look in that.
Okay.
Yeah, I can present those to you, Michelle.
Yeah, that'd be great.
Is PharmCAT available to other institutions?
Okay. Yes, PharmCAT is available. So PharmCAT is in a beta version right now, and we just got funding to make this more hardened code and get better. So right now it's available as a beta test case. It does not, it needs to be updated with a few more Cpic genes, and it needs to be updated to where it can run in a batch, so you can do hundreds of patients at one time, because right now it runs one patient at a time. So it is available. I think it's pharmcat.org. If you go to pharmcat.org, you can find information about it and the current status. Like I said, we just got 3 years worth of funding that we're looking forward to updating that code and making it an official release, not a beta release. That will be happening in the next couple of years.
Great, thank you. The next two questions are related to PharmCAT as well. Are any of these PharmCAT set up for APIs, and is there a fee associated with PharmCAT?
Okay, so there's no fee. It's NIH-funded, publicly, freely available. And as for API, so part of our funding is to be able to integrate PharmCAT, the PharmCAT tool, with EHRs. That is a I'm sorry, I'm blanking, a part of our grant proposal. So we will be doing that. Currently, that does not exist yet for PharmCAT. So right now, it just takes in the VCF file and it gives you an HTML or PDF report at the end.
Great. One more question in the queue. Has Cpic or PharmGKB developed an add-in set of probes that can be used for boosting exome coverage?
No. So we don't have, at PharmGKB or Cpic, we don't have that kind of wet lab association where we're developing probes, et cetera. However, the AMP group, led primarily by Vicki Pratt, there's an AMP PGX working group that I'm associated with, where we published some papers about what it's kind of called a must-test SNP list per gene. What alleles you really need to have on a panel if you're going to do that, or be looking for an exome. They don't necessarily have probes available, but as part of that work, we do point out reference sequence reference material availability. And so that group might have more information about particular probes to amplify particular variants.
Okay, any other questions? This has been a fantastic overview by Michelle. Thank you so much for your time.
Great. I'm happy to do it. My pleasure. Thank you very much for your attention today.
Thanks.
Thanks, everyone.
Thank you.